Hello, my name is Hayden Braun. Today I'll be talking about epigenetics, a fairly new field in biology. So, what is epigenetics? Epigenetics is how the environment of a cell can affect how our genes are expressed. Things which can create such an environment can be stress, our diet, and our sleep schedules. The effect of epigenetics can be severe, such as cancer, diabetes, and mental disorders. Now, before we jump into how epigenetics works, let's recall that DNA is compacted into the nucleus of a cell as a chromosome, and part of this packing process is DNA winding around a histone proteins, making a bead-like structure. These proteins can then move along the DNA to expose sections. If this section were to be replicated, nucleosomes will surround the region of DNA, and RNA polymerase will begin the transcription process. This is where epigenetic factors come into play. The first epigenetic factor we will be looking at is acetylation and deacetylation of histones. The premise behind this process is that histones are partially positive and DNA is partially negative. Now, for a moment, let's imagine that my finger is a histone and this string is DNA. If the histone were to be acetylated, the positive charge would then be decreased, lessening the attraction to the DNA, causing a slight uncoiling effect on the DNA. This will help the RNA polymerase to transcribe the genes, allowing them to be expressed. On the other hand, if the histone were to be deacetylated, the positive charge it possesses would increase, causing the DNA to coil even tighter, like so. This prevents RNA polymerase from reaching the DNA, hence halting the transcription process and silencing the gene. Though this factor does halt transcription, it is not the most prominent. That title belongs to the process of methylation. There are about 3 different methylation processes, but only one of which are seen mostly in eukaryotes. That is cytosine being methylated to M5 cytosine, or 5-methylcytosine, as seen here. This happens in areas of high cytosine and guanine density, called CpG islands. When this happens, the cytosine can no longer be recognized, and therefore cannot be transcribed. This is like when you are reading a book and there is noise, a lot of noise, causing you to lose your place or not comprehend what you're reading. This methyl group on the cytosine can be thought of as the distracting noise, causing the polymerase to not recognize what it just read, causing the gene to be silenced. For example, if I were a polymerase reading this line of DNA, I would read it like so, as I just transcribe it. ATGTCGCGATT. Easy enough, right? Let's see what happens when I try it again, but it is methylated. ATGTCG. Wait, I don't know that one. I hope it's not important. And then it would continue on, and this gene would not be expressed. However, there are other consequences which follows 5-methylcytosine, as it can be deanimated, which is the nitrogen is replaced by oxygen, to thymidine, causing the CpG site to be mutated. This increases the chance of a permanent genetic mutation. Now, you might be wondering, why worry about epigenetics? Well, as we discussed earlier, it can be the cause of various diseases and illnesses, but it also can be passed on to your offspring. Now, these changes may not be permanent, but they can be drastic. For example, in a 2018 study, epigenetic changes were observed among child abuse victims. This experiment was done by collecting cortex tissue from 3 groups. One group from people who've died suddenly with no medical or psychiatric history. This was the control group. One group were from people who have committed suicide from depression. The final group from people who committed suicide with history of child abuse. Differentially methylated sites on the DNA was then observed by looking at high-density CG areas. They then used bisulfite sequencing, which uses restriction enzymes on the DNA to produce sequence-specific fragmentations. Cytosine will be changed to uracil, but 5-methylcytosine will remain the same. The results showed that those who experienced child abuse showed both hyper and hypomethylation, compared to the other two groups, in 3 regions which relate to proteins and myelination. Myelin is a protein associated to the speed of firing impulses, meaning their stress-inducing environments cause these proteins to be silenced, slowing their impulses and their increasing the risk of depression. However, these changes were not seen in the depressed group. This suggests that these changes to myelin are directly related to child abuse, showing childhood environment will affect myelin transcription for a long term, contributing to impulsiveness, aggression, anxiety, and depression. Further testing with rodents shows that when the rodents were isolated, they too suffer from decreased myelination. And once they were reintroduced, they did not recover. So, are epigenetic factors really temporary? Yes, but the factors introduced during early life will have a higher chance at making a lifelong impact. As it has been seen, a wide range of causes, epigenetics has a wide range of causes and effects, from substance abuse of drugs such as cocaine, meth, and alcohol, increased acetylation of the histones to high levels of stress, resulting in change in mood and disease. But is there anything we can learn from epigenetics? As it turns out, by studying this field, we can obtain a better understanding on how diseases develop and a possible intervention. In fact, possible epigenetic drugs are being explored to help counteract the very diseases that they may cause: cancer, autoimmune diseases, and neurological diseases. By further studying this field, we may continue to find the cause and possible and possible solutions to many other illnesses.